Soundstripe.
As we're settling in, let's just go ahead and get started. Thanks everyone for making the time, for showing up. I think you won't be disappointed. All right, so let's start with the following. CU Denver is with the following acknowledgement, right? Really important. CU Denver is on the traditional territories and ancestral homelands of the Cheyenne, Arapaho, and Ute nations. Let us acknowledge the painful history of genocide and forced removal from this territory and pay our respects to the diverse indigenous peoples still connected to this land. Let us give thanks to all tribal nations and the ancestors of this place. And then within that, right, the important thing to always mention, be aware being on this campus, right, the histories and legacies of displacement and removal that have occurred here. And there was a year ago, there was like an event, so where they describe and then look at the history, and they have like really nice videos and then other things. So I highly recommend that, since we are all on this campus, that we know about that. All right, so then just a little bit about the Dreyfus Lectureship for undergraduate institutions, right? So last year we were granted as a department of chemistry within the college, right? So this prestigious award to host a renowned person, speaker, right, to come to campus. And the main thing with this, or the main aspect of this, is basically that the speaker or this person, right, interacts with students, or as much as possible, certainly more than in a regular seminar series, right, that takes place on Fridays. So that's part of it. So there was a group of students that came together, right, and then they started through a process to basically pick or come up with names or pick the candidate, right? So after that, Professor Garcia-Garibay was the selected candidate, and then luckily for us, right, so it all happened smoothly. He agreed, had the time, found the time to come and visit our department. And so that's what's happening today. As part of this lectureship, right, so there was also support for students to carry out research over this summer, so that occurred, right? So and that is important to know, that with support of the Department of Chemistry and CLAS, also our college, right? So this was expanded to 4 students. So it's original for the Jean Dreyfus Lectureship supports 2 students. So now we were able to support 4. So we are thankful about that. And so as part of this series, right, so something that's different is that there is the public lectureship, right, which we're going to hear about today. Tomorrow there's going to be a focused chemistry lectureship, right? And what you'll see is that it's going to be on 2 complete different topics, right? So if you don't like today's, you like tomorrow's, or something like that. No, but it's like very, very broad, different research. So you'll see that. It's in both areas, right? Incredibly interesting. So you'll see about that. And that's basically what you see on the flyers, right? For the chemistry lectureship, and then today public lectureship. Then another activity that we put together, right? Students put together, is basically a poster session. So that will take place tomorrow at 3 to 4:30. There's going to be, some, I wouldn't call it appetizers. We're just like, what is it called? A pop cart. Not pop cart. What do you call it? Popsicles. A popsicle cart. Yeah, for the heat. So just so for the first, so get there early so that you can get 1 or 2 or 3. Anyways, that happens at 3 to 4:30 is going to be outside, so hopefully it's a nice day. And so, right outside the Butterfly, I don't know if it's called Butterfly Pavilion, but whatever it is, where the Butterfly is, the coffee shop in the Science Building. So just go there outside, that's going to take place there. And so without further ado, I'm going to give space to Emily, who is going to introduce our invited speaker, and then maybe we can, well, you'll set your computer.
Hi. To announce our speaker of today's lecture, Dr. Miguel Garcia-Garibay is a distinguished academic leader who continues to leave an incredible mark on the world of higher education and scientific research. Dr. Garcia-Garibay received his bachelor's from the University of Michoacan in Mexico, his PhD from the University of British Columbia, and his postdoctorate fellowship at Columbia University before joining the faculty in the Department of Chemistry and Biochemistry at UCLA. As the Dean of the University of California, Los Angeles, he has not only assumed a position of administrative responsibility, but has also continued to demonstrate a commitment to the pursuit of knowledge and innovation. With a career marked by scholarly excellence, a passion for education, and dedication to fostering an inclusive and diverse academic community, Dr. Garcia-Garibay is a prominent figure of one of the nation's most renowned institutions. With that being said, I am happy to introduce Dr. Garcia-Garibay and his lecture on crystalline molecular machines.
Thank you, Emily. Can you everybody hear me okay? Okay, perfect. All right, so while I set up, I want to really thank you all for the opportunity to share with you some of my group's thoughts about molecular machines. Okay, let's see. So the Jean Dreyfus lecture is really special because it's only awarded to some of the best undergraduate institutions, and the fact that you got it, I think, is pretty significant. Let me see. I'm going to have to do this again. Excuse me. Okay, now we should be in business. Good. Should we dim the lights a little bit?
Yeah, let's do it.
And maybe. Okay. Okay, perfect. So the Dreyfus Foundation, as you might know, is a philanthropic organization of the Camille and Henry Dreyfus Foundation. And I have to say that because higher education today, universities and research in higher universities relies so much on philanthropy, so state governments really stop supporting us. And now we rely a lot on public support through foundations and private support. So I say that because some of you will be very successful, will make a lot of money. So when you do that, think about see you Denver and give it back. So hopefully you're having a wonderful experience here, and then you can make a change in making sure that this becomes always a better institution. So that's one of the things deans have to do. In my job of dean, I have to promote, the sustainability of our department, our campus. So this is my title today. It's an ambition title, crystalline molecular machines. So I'm going to do my best to tell you what we're talking about. I know this is a public lecture, and so I'll try to really share with you the general concepts and make it as accessible as possible so that some of you might be excited to get into this particular field, okay? As a general outline, if you see here, we start with a quick peek. So what are crystalline molecular machines? So what am I talking about? So first of all, I want to give you an idea right out as to what we're thinking about. So what's a crystalline molecular machine? Then really we go to the real molecular machines that are amazing. And many of you study biochemistry and you know about the molecules of life. That's where there is a lot of really exciting machine-like function that makes life possible. We transition into artificial molecular machines, and I will explain some what I think are the key concepts that play a role in the field of molecular machinery. Then why crystals? And then we will explore a concept that was developed at UCLA in my group by my students, the concept of amphidynamic crystals. So think about what amphidynamic crystals might be, and we'll explore that a little later. We transition into how do we measure motion and machine-like function in crystals? We need to understand how do we actually know that molecules are moving within crystals, right? And then we end up with some examples. So let us start with a simple machine. We're going to select a couple of navigational machines, relatively simple. We have a gyroscope on the left and a compass on the right. And in the center, we have a cross-section of those two structures, right? Those structures have completely different purpose. So a gyroscope relies on rotary motion that carries angular momentum. And you all remember about the conservation of angular momentum, right? So that's the principle of a gyroscope being a navigational device. You can be in outer space, and the gyroscope, if it doesn't change anything, it doesn't exert any thought, you know that you're in the right direction. A compass, on the other hand, relies on a magnetic needle that will search the strongest magnetic field. Normally that would be the Earth's, right? If you notice what structures rely on a framework, there is an axle that joins both sides of the framework. For some reason, I cannot make this work. Let's see, Marina, maybe you can help me out. Oh, you have to turn it on. I don't need that, just a part there. Once you turn it on, right. So, in this case, this is the frame, and the axle goes from one end of the frame to the other, and then there's the rotary mass in the center. In the case of the compass, we have the needle, right? So they have a similar architecture, and when you see how do we materialize this cross-section with a molecule, I've shown here a molecule based on carbon, as you can see, where I follow the colors. So blue is the frame, and on red we have the axle and the rotator. Now in this particular case, I have two fluorine atoms so that we have the equivalent of a magnetic moment, but instead of magnetic moment that will interact with a magnetic field, we have an electric moment that will interact with an electric field. So just like the compass will align to the strongest magnetic field, an electric dipole will align with the strongest electric field. But there are similarities. But we're talking about crystals, right? So what we're talking about is of structures that are more like that. So we will have a collection of gyroscopes or a collection of compasses. And then you might say, well, so what's the advantage of that, right? So how does that make it into a crystalline molecular machine? So there is a little bit of a hidden secret about when you bring too many things together. When you have many things together, many gyroscopes that have individual angular momentum, or many compasses that have individual magnetic moments, they can interact with each other, right? So here we have a 3D representation of what we're talking about, so that you have visualize a little bit more what those molecules might look like. Here we have, this is a really interesting picture, look at this. The arrow here indicates where the magnetic north of the Earth is, and that isolated compass is doing this job, right? It's telling us where the north is. So what do you notice about this one and this one? They're completely ignoring the magnetic north because they're working together. They're working as an ensemble. They display a property that is called emergent phenomena, something that it only occurs when you have a collection of objects coming together and working together. Now, when you have an object like that, now we have a macroscopic polarization, because now all of them generate a magnetic field that is stronger than the magnetic field of the Earth. And if I put more and more and more, I make a stronger and stronger magnetic field. At some point, I can actually make a magnet. But if you notice here, so this is if these were molecules, these are very cold molecules because everything is static, right? But what if we put some heat? Well, if we put some heat, then we can actually activate some of those molecules, as you can see here. So this is a cycling movie, but what you see here is we start there, And then I come with a toy magnet in the back, and I start to play with one of these. And you see, I play with this, but I can affect everything in the ensemble, right? That's another emergent property. So this is what we're after. Another representation of that is shown here. So that's kind of what a crystalline molecular machine will look like. So this is a machine unlike any one we might have, right on Earth, but it's one where you can have molecules work together like that. You can actually have an opportunity to manipulate electric fields, magnetic fields, and electromagnetic fields. So it's an opportunity to have a crystal, right? So this might, when you make a crystal like that, might look something like that. And that crystal will give you an opportunity to interact with matter in different ways that you couldn't otherwise, by virtue of the fact that there are properties of these molecules that have their motion internally. So we go from a macroscopic machine, an instrument in this case, say a compass, and we make a crystal that contains these molecular compasses, and we begin to explore the properties of those. So that's kind of what we're looking at, right? So our goal is to design and build molecules that have desirable functions as a result of their internal motion or ability to change shape. That's going to be more or less our goal. And there are many other incentives to look at molecular machines. So chemists over time have looked at bevel gears, spur gears, gear trains, and you see here some animations of the type of structures that we have worked on in my lab. So we've made molecules like this, like that, and like that, which is the same as this. So there are similarities, there are interactions, there are possibilities. And what crystals do is that crystals make all of this be ordered. They can be organized. Crystals help us reduce entropy. So that's kind of the peek into it, right? Now, I mentioned that life really is, the machinery of life is incredibly interesting. I don't know if you've seen this book, The Machinery of Life by David Gottsell. In the front cover, there is this image of an Escherichia coli bacteria. And clearly there is a lot that is going in terms of molecules that have machine-like functions that is really, truly interesting, right? So the machinery of life goes from simple bacteria to complex organisms to the brain. And this is all molecules doing something, right? This is biochemistry, this is chemistry. So there is a lot to be learned from biology. So I like to make the connection between biomolecular machines and artificial molecular machines. So, I selected this image just to remind ourselves that we coexist with microorganisms, so these are the simplest species. If you were to collect a sample, say, from your skin and put it on a Petri dish, you grow bacteria, and it might look something like that, right? Many different bacteria, and you'd be surprised that, I just took a shower this morning, what happened, right? Well, even if you took a shower, you still have some bacteria in your skin. As you know, we coexist with bacteria, something we call the microbiome, right? And it's actually healthy for many, many different ways. So if you take one of those colonies and dilute it and grow it, then you get a distribution of colonies like that. You look under the microscope and you get a bunch of bacilli like this. And this is basically the image that we saw earlier. So let's go back there. I love this image from David Gottsell. This is actually an artistic representation of what a bacteria might look like. And I just love it because it really opened my eyes as to what a cell might actually look like. It's crowded, it's messy, right? So this is not an ideal 1 molar solution of anything. Right? This is very complicated. So what you see here is we have our bacteria, and there is this structure here, which are called the flagella. And we can find movies on YouTube of flagella bacteria swimming, and we see the flagella in action. So what you see here, the flagella actually come all together to one end of the sail, and like a corkscrew, they help the bacteria move in water. Now, bacteria feel water as a very, very viscous fluid. So for us, we're big, we're heavy, so water feels very fluid, very easy to move on. For bacteria, it's like if we would have to move through molasses. And that's why they use a corkscrew to move through a very, very dense medium, which is really interesting, right? So if we now look at the color section here, so that we can see the flagellum up close, we come to this image. And I can look at this incredibly complicated structure, right? So these things are, proteins. We see phospholipids here. There is a membrane. Here is the flagellum. This is the flagellar motor and the flagellar filament. And so this is a molecular machine. This is a very complex molecular machine, right, that nature developed through evolution. In very rough terms, the way it works is that there will be charged atoms, ions, that go from this end. They pass through the bacterial motor, and you can think of it as a current, right? So there's a current, and that brings some rotary motion that is transduced to the flagellum, and that's what happens here. So this is an amazingly interesting and complicated molecular machine. Right? It's all made of many different proteins, and you can think of it as a long polymer also. And ions are going through, and ions are coming through the other end of the flagellum. So you can also think of it as a propulsion system. Now, if you look closely, again, I see this, to me this is really fascinating, right? So here we see some ribosomes making some proteins, right? Another really amazing molecular machine, right? What you're taking, the information from RNA and from that transfer RNA to make proteins. But the one I want to focus on today is this little guy here, right? That is ATP synthase, right? This is the protein that basically makes the molecules of energy that we use for everything that we do. And there is this beautiful image that you can find again on YouTube. And so this is an amazing rotary machine, and the way it works is also that there will be protons, charged ions that come from one end of the membrane. That is a high density of protons, so a high pH, to the other side. So there is a gradient. They go through, they actuate the protein, and what you see happening here is that these things that are coming in are ADP, adenosine diphosphate, plus inorganic phosphate to become ATP. It's synthesizing ATP. And every third of a turn makes one molecule of ATP. It's a beautiful machinery. And this gives me an opportunity to give homage to one of the people who had a very important influence on me, Professor Paul Boyer, who was my colleague at UCLA, came up with the primary mechanism of ATP synthase. This is an amazing machine, isn't it? It's beautiful. And I wish, we could do something like that artificially, but we're very, very far from that. Now, let me talk about another machine really quickly and very superficially, the machinery of vision, right? So we see a tree here, and of course you know that what's happening here is that there is a tree that you see, and the photons that come bouncing off those trees that pass through the lens of your eye are reflected in the back of your eye here. And we go to the next image, and we see that there is a construct here of cells, cones and rods, that where the light penetrates. The cones and rods have this protein, rhodopsin. And if you squint really hard, you see that there is something here kind of in purple. So this is a large protein, and you see all these helices here. And what's in the middle there is this molecule, retinal. Now, retinal normally is a bent molecule, as you see here. And when a photon arrives, when light comes in, it undergoes a change, a chemical change. It goes from being bent to being linear. And that actually becomes, transduces energy so that there is polarization in the membrane. But what I want to say here is that molecules that change their shape, of course, we call them stereoisomers, really are the mechanism, the trigger that causes a lot of machine-like function. So many things that happen in life can be reduced to a conformational change, a configurational change. And in this case, cis-retinal that is bent goes into transretinal. That causes changes in the membrane that effectively will cause a polarization of the membrane. And the process of changing structure or stereoisomerization will trigger an electrical signal that goes onto the optical nerve and to the brain. So the key concept here is stereoisomers and stereoisomerization as an important thing. Right? We said that design and build molecules that have desirable functions as a result of their internal motion or ability to change shape. So this is the ability of the molecule to change shape as a photon comes as an external stimulus. In this case, we're talking about stereoisomers. So, our organic chemistry serves us well to understand many of these processes. I also need to recognize that, because molecular machines have been recognized with the Nobel Prize in 2007, when Professors Jean-Pierre Sauvage, Sir Fraser Stoddart, and Ben Feringa were awarded the Nobel Prize for the design and production of molecular machines. So this is the citation in the Nobel Prize website. And if you look at the website, also they have these images, right? So Stoddart developed, among other things, something that is called a tiny lift. So you see here, the analogy with the lift is pretty evident, right? So there is these 3 stations here that go from the bottom to the top. So there is a function, there is an analogy, and that is the ability of design a molecule that resembles a macroscopic machine. Professor Sauvage developed artificial muscles that are molecules that look like that. So these are molecules that basically can slide past each other, become compact or extend like a muscle does. And this can be controlled artificially through by including one type of an ion here and another type of an ion there. So there is shuttle of this, and that causes the stretching function. And then Professor Feringa developed a molecular motor that this image is intended to represent that this is a 4-stage motor. And you can compare that with this image, right? So this is again a representation of a internal combustion motor. And I think now you're thinking that, wait, these are isomers, right? This is an isomer where the 2 internal pistons are in or out, or in or out. So these are different structures. Changes in structure that produce a function is what makes up a machine. So there is a strong connection between structure and function and motion, and that's what happens in all of these types of molecules. And perhaps one day we can make things as complicated as this. All right, so let's see. Again, just to bring that in, right? Harping on the fact that we want to look at desirable functions as a result of internal motion or ability to change shape. Now, what about completely artificial macroscopic and molecular machines? So there's a really interesting difference that I think is critical. So this is a representation of a bevel gear, right? So depending on how many cogs you have here, so you have a number of rotations that is going to be a multiple of the number of cogs in the little one with respect to the large one, right? So the thing about macroscopic machine is that you need energy to transmit motion from one axis to another in this case. So you have to put energy, if not, they're static, right? So if I remove whatever energy is moving that, it will stay static. Now, when you look at molecular machines, what you find is that, let me go back. Well, it's supposed to be a, let's see if I make it work. All right, there it is. So they never stop. Molecules never stop. Even at 0 Kelvin, molecules keep moving, what we call 0-point energy motion. So it turns out that molecules are different. So what we need is we need to control the motion. So we need energy for them to stop moving randomly. Molecules move randomly with Brownian motion. So we need to do something special. We need to break whatever symmetry exists so they can move only in one direction and not in both. Let's take a look at another machine, right? How about a wristwatch? And so how many stereoisomers do you think we have in a wristwatch? So I'm calling stereoisomers. Let's find. So can you see the needle? Right. So every time the second needle moves, that's a new isomer, right? It's a different structure in space. So you think about it, so we need a number of isomers in this structure in order to read time. So we are decoding the structure of isomers to tell time. In fact, to encode 12 hours, we need of course 60 seconds for every 60 minutes for every 12 hours. So we need 43,200 stereoisomers. So you need to start to think as a chemist to translate the language of macroscopic machines to biomolecular machines to artificial machines, right? So that's what we're trying to do. There is another thing that is quite interesting. Here is again our watch, and so let's assume that this is a molecule. So as a synthetic chemist, as a chemist or biochemist who's designing something, the first thing you need to do is to have an idea. So your idea is going to be some blueprints, right? So you need the blueprint. So I want to make that, as complicated as it may be. And then once you have an idea, you have to go to the lab and you have to make every single component. So you go to the lab and select the materials to build every one of these components. So this could be different molecules that will come together to make a supermolecule, like a protein aggregate or a supramolecular aggregate, right? And if you think about it, let's say we have 100 different components here. So as a chemist, one of the things that we need to think about is how many variables do I need to tell the structure of 100 different components? And what do we call that? Well, if you've taken general or physical chemistry, you might remember, we call that degrees of freedom, right? So we need 3 translational degrees of freedom for every object, and 3 rotational degrees of freedom from every object. So we need 6 degrees of freedom for every object. So if we have 100, we need 600 degrees of freedom. And if we shake them in a box, they can go crazy, and you'll have a hard time describing the position of every component. So what's the next thing we need to do? We need to assemble our components, right? And that happens in nature, and that can happen also artificially. So they all begin to assemble in an expert manner, and then what you have is that our machine, one machine has still 6 external degrees of freedom and 1 degree of internal freedom. So what we've done is we've taken a large level of complexity, many components that independently don't do anything, put them all together, and that's called a reduction of entropy, right? So we're reducing the entropy of the system in a very significant way, and guess what? That's what crystals do. Crystals take molecules that have a huge amount of entropy, and then the molecules are assembled in the crystal in a way that every molecule is precisely located in a position, and there is a tremendous reduction of entropy. So we're beginning to make a lot of interesting connections here, right? So we already said, okay, so we have, we said 43,200 stereoisomers. You say, well, I don't really count seconds, so maybe you want to say, well, maybe then you need 60 times 12 or whatever, but whatever, you need a lot of stereoisomers. We need something else, right? So in chemistry, we need one more parameter, which it considers the energetics. So how are these going to happen? So here are our stereoisomers. And now what we're going to have is we need energy, right? So we represent energy by a potential like this. And basically what we're saying is, well, this isomer spends a little bit of time there, and then it moves to the next isomer, spends time there, moves to the next isomer, spends time there, and it keeps going, right? And it will keep going from one isomer to the next as long as there is an energy gain. In this case, we're taking the spring energy, and we're transducing the spring's energy through gears and ratchets, and we move from one isomer to the next, to the next, to the next, and so on. Right? And as long as we have energy to transmit that motion, we'll be exploring those isomers. So that's what we call, potential energy surface or energetics. So in the case of a macroscopic object like a watch, we need to think about inertial forces and friction. Now what about a molecule? Let's go back to our ATP synthase molecule, right, in a different representation. So this is not something that we think often, but it turns out that if we had no energy difference between, say, one side of the potential. So here we start with the protein plus ATP. This is actually, I guess it's a phosphatase. So stays here, moves and stays here, and moves and stays here, and moves and stays there. But you see, if the energy of these two is the same because there is no difference in chemical potential, it's going to be a random motion. It's going to be to the left, to the right. One times it makes, say, ADP plus, one time is a phosphatase, the other one is a synthase. It's totally random. So molecules are random. So what happens then is that this needs to be coupled with something else that provides the energy to make it not random, right? So now what we have is we have a large chemical potential, and in this case what it means is that we have a lot of protons on this side of the membrane. And those protons need to have accumulated by some other mechanism, and then we can continue making, every time having an enzymatic event, right? And that's how our machine is going to be working. So the chemical potential here just has to do with the relative concentration of ATP or ADP, and that's going to determine if it goes one way or the other. But if you have the same amount, it's going to be a random Brownian machine. So that gives us hope, right? Because if we make a random machine, we can still figure out how to bias it in a way that it makes it unidirectional and useful. Now let's think about so you're asking, okay, but why do you want to use crystals? So why crystalline molecular machines? I already told you the first thing, right? You can have as many as 10 to the 20 molecules, and they're going to be identical, right? So a molecule in this part of the crystal or in that part of the crystal are identical. Anyone in the crystal are identical to a molecule here or there or there. So they're all identical. We have a large reduction of entropy. So that's an incentive. It's an easy way, a simple way to reduce the entropy of a system. The other one is that we can encode function in structure. So if we want to mimic a gear, we can mimic a gear. If we want to mimic a gyroscope, we can mimic a gyroscope. If we want to mimic a four-fold cyclic gear, we can mimic that. So today, synthetic chemists can if you visualize a structure, you can picture how to put those components together in an artificial manner. So that's another incentive, right? Another incentive is that, well, crystals can be very little. So you see this is an SCM picture. This is a 100 nanometer bar. So 100, I guess those would be billions of a meter. Very small. Or if you see here in the Nika caves in Chihuahua, Mexico, there are crystals that, if you look carefully, this is a person. This is perhaps a man. And look at the size of these crystals, right? So some of these crystals can be as large as 20 meters. So crystals can span from a few nanometers to many meters, and that's also potentially interesting. And crystals can actually actuate from the molecular scale to the macroscopic scale. So let me tell you what's happening here. What we have here is a crystal where we're going to put the laser right there, and then a reaction happens at that point, and that reaction generates a lot of energy and strain, and then, like a ballistic missile, breaks the crystal in two. What's happening here is that the light is coming from the top. So the crystal is straight. When the light comes from the top, it causes a chemical reaction on the surface that makes the crystal bend. So if you use that principle and bring it here, you can actually use a crystal and a laser to move a large gear. And this is macroscopic, right? And in this image, what you have is that when we get it back, you have a crystal in a hot stage, and when it reaches a certain temperature, there is again a chemical change in the crystal, and that transduces energy into, boom, a crystal jumping. So what that tells you is that you can actually interface a human being, one of us, can talk to molecules by using external stimuli. So, I can pick a crystal, I can do whatever I want with the crystal, and I can do something to that crystal and talk to those molecules to do something at the macroscopic scale. That is another incentive. All right. So let me introduce you the next concept that is going to be important. So what you see here is a replica of an adding machine designed by Leonardo da Vinci. And I look at this and I find crystal-like order. There is periodicity. There is many gears here. It's dense like a crystal. And what I say, what is special about a machine? Well, that is, it brings some amount of order and some amount of motion, right? So in this particular machine, you set up the knobs and crank a crank, and you can get the sum of two numbers. So it's an adding machine. So this is a smart object, right? Because it can do arithmetic. So it's some motion and some order. So then that means that machines bring the conjunction of motion and order to different degrees. So ATP synthase again, right? So has a lot of structurally encoded order. Every single of these protein units plays a role and is precisely ordered. It's ordered with respect to the membrane. So there is a lot of order, but there is also structurally encoded motion. So the structure that encodes the order also encodes the motion. So machines, then molecular machines, are again the combination of structurally encoded motion and order. We're going to call those molecular machines. The next thing we're going to do is, so what does nature gives us in terms of order and motion? And so what we're going to do is going to put everything that we know about. A lot of order and very little motion, regular crystals. If you want a lot of motion but no order, you can have a liquid, right? Now, there are plastic crystals and liquid crystals that have increasing amount of motion but more and more disorder. And then you can have glasses where there is no order, nor motion. So what you have here is that this is really, in a way, it's a new state of matter. It's a different type of condensed spaces. And we call that amphidynamic crystals. So amphidynamic crystals are crystals that combine the order of a crystal by having frameworks or lattice points that are perfectly ordered and static, but also have components that can be mobile, that can rotate, can translate, that can have some motion. So they have structurally encoded order to make a crystal, but they also have a structurally encoded motion for something to move inside. So what we're going to explore is, so what are the potential structural solutions? Well, one is that you make crystals where you have a lot of empty volume, right? So I'm showing here, for instance, a pendulum. In this case, I'm showing, this is called a metal organic framework, where we have, you can see that there is all of these beautiful atoms, because it's actually moving. So we take an X-ray structure, but everything is moving in this crystal, because it can move in free space. So one solution is what we call free volume motions. And those are metal organic frameworks or covalent organic frameworks. What other solutions might we have? How about volume conserving motion? So we can have objects that as they, say, rotate, if there is a rotation, there's no change in its shape. And that's how most machines are, right? When you look at a gear train, it's not moving, right? Everything is rotating, but without changing the shape and without really requiring more space. So this can move without requiring any space. So we call that volume conserving motions. And I'm showing you the molecules that we've synthesized to solve those structures. So these are actually molecules that have been synthesized. This is, of course, a fake animation, but the molecules are actually real. The next is correlated motions or gearing, where the rotation of one will influence the rotation of another. Right? Those are molecular gears. These are the most challenges because they require a lot of design. Now, the molecular gearing can occur through basically steric interactions, or they can also occur through dipolar interactions. These are supposed to be little dipoles, or like a little magnets. If you move the one in the end, you can move every one because there is information encoded that can be translated. So these are the structural solutions. So we can have free volume, we can have volume conserving motions, or we can have correlated motions. And that's how you can design motion in a crystalline solid. The next question is, so how do you measure it, right? Well, measuring rotation and motion in solids is actually not that hard. It's actually relatively easy. All you need is a high field NMR, as I'm showing here, and then you need a sample that can be either single crystals or powders. If you're using powders, you can put them in a rotor. This is a device that rotates very fast inside the magnet. And by measuring the spectra within the magnet, you get a structural information about motion. So this can be depicted in this way. So let's assume that we have three regimes, molecules that have really no space, and we can only have a local oscillation. And the spectrum, in this case, we're looking at deuterium NMR. The spectrum looks like this. So it looks like a Batman kind of thing. And that means that it's static. Now, what if you have a little bit more space? So if you have a little bit more space, as we have here in the center, now it can flip from one side to the other, like flipping a pancake. And the spectrum changes, and the spectrum looks like that. Now, if it goes continuous, if it is continuous rotation and looks like that, the spectrum sharpens. So all I need is to put the sample in the NMR machine. I collect the spectrum. I look at the spectrum, and I can actually, from the spectrum, draw the information about the motion. So it's very nice. Now, this is not the only way to do it. I can also use different techniques. So, for instance, in a molecule such as this, the part of the center, which is called diamante, is very fast. It's so fast that it's hard to measure. So what we're doing here is we measure a technique that is called spin lattice relaxation. And we get data where we get the relaxation time as a function of temperature, and we get a curve like that, and from that we can determine how fast this is moving. We have to do a little bit of math by using equations such as this. Or if we want to look at the rotation of the benzene rings shown in red here, those are very slow. So we can use a different technique. This is called a variable temperature line shape analysis. And again, we get, as the spectrum changes, we can tell how fast is it moving. So we spend a little bit of time learning on how to use these techniques, but we can tell how fast this is happening within the solid state, which is very useful. So let me give you an idea. So this is actually very sensitive. So we made this molecule. So this would be what we call the stator, or the frame in blue, and the rotator in red, right? So we measured that, and it was static, no motion at all. The crystal was very dense. So we said, well, maybe this is just too rigid. So what we did is we removed one atom here at the top, and we make this molecule. And this molecule undergoes 800 rotations per second at room temperature. And if you do simulations, it looks like that. So it actually looks like a little machine, right? So we call this crystal fluidity because the crystal is making room for the rotation to take place. So if we change from a flat rotator, like a benzene ring, and we make a bicyclo to octane this cylindrical shape, now we have 110 million rotations per second. And it's otherwise the same, right? So this part is the same. The only change is that we change from a flat to something that is more cylindrical. And of course, flipping a flat disk is more difficult than flipping a cylinder. Makes sense. So in this case, we have what we call volume conserving motions, right? So here we have what we call crystal fluidity, and here we have volume conserving motions. So we've been learning so much about motion and order in crystals because none of this was known before we started working on it. So we can get back to our original idea, right? So what about our molecular compasses, and what about emergent phenomena? So what you should notice here is that when we have dipoles aligned in a square net like this, if you turn this to the right, that will move that, and that, right? So everything is correlated. And if you move it a little further, then you move it to that. And if you see, this structure is the same as that structure, except rotated 90 degrees. So it means that everything is correlated. So in this case, we're moving the one in the center, and everything is moving. In this case, it's a bit more random because we're not doing it with a homogeneous field. We're looking with a local field. But what if we have a trigonal array? So if we move this one, everybody follows. If we move it a little more, everybody follows. So this is an opportunity to rotate the orientation of the dipoles. And this, as I said earlier, has an effect on interaction with light, with electric fields and magnetic fields. So there could be new materials that we can develop from that. So the structure that actually was successful was this structure. So in this case, I'm going to have you pay attention just to this part in yellow. Right? So this is the rotator. This is the framework in blue, and the rotator is in yellow. And there is this is the needle, where the direction of the fluorine atoms, right? So the first thing that you probably notice that when we get a crystal structure, where are the fluorines? They're everywhere, so because they're rotating very fast. So the fluorine atoms are just going around it, and so this is a crystal structure that we're obtaining, where we see the distribution of the fluorine going around and around. But what happens is when we cool it sufficiently, all of this becomes ordered. So all of the dipoles become ordered and adopts an orientation like that. It's called an icing lattice, and I'll show you how we detect that. So this is what's happening. It's random, and then we lower the temperature, and then it begins to organize. And then it settles. So spontaneously finds a way to organize. And we can detect that by measuring capacitance. So we measure the electrical response of the solid, and we can tell that when this peak happens, it's because it became all ordered. So this is the first example of a rotary array of dipoles that spontaneously find a way to organize themselves. So this was a significant accomplishment. So let me show you maybe one or two more examples, and then we wrap it up. One example that I wanted to share with you is, again, we want motion and function, right? So there is this molecule. This is called a salicylic aniline. And if you notice the difference between this and that isomer, right, we have two isomers, is that here we have the hydrogen attached to oxygen, and here we have the hydrogen attached to nitrogen. Now, this molecule is colorless, and this molecule is orange. So, when light comes in and excites that molecule, it becomes permanently orange. So, this is an equilibrium where colorless and orange go back and forth, with the colorless being preferred. Now, when we use light, we make this one where the hydrogen is now far and it cannot go back. So that hydrogen cannot go back there to make that. So we can trap this. So this is the first machine-like function. But we want to put it in a crystal. So if you put it in a crystal, it's not efficient. This is not a good reaction, because it's very hard for that nitrogen to move down and that carbon to move up. So this is an isomerization reaction that is not likely, it's not possible. So we needed to engineer it. We need to make a machine around it. And that's what we did, right? So schematically, you see that we have our molecule in the center now, and we created a case where the molecule is. And that's the actual structure that we made. So now this is going to act as the frame, and this is going to act as the axle, and we're going to have a reaction. And if we're successful, our crystals are going to change from yellow or almost white to orange. And I have to tell you that we use a lot of chemical synthesis, and that's the only slide I will show. But I want to acknowledge the fact that if we didn't know how to make molecules by putting together, these nice structures, we couldn't do anything. So there's a shout out to good chemists. So let me go back. So this is the result, right? So you can look at here. So we cover this part of we put the solid in here, cover this part with tape, and expose that to light. You can see clearly, right? And you also can see how the spectrum changes in the region where we see that the yellow appears, and it's not until we use light of a different wavelength that we can bring it back. But we were able to make basically a molecular machine where the structure facilitates rotation and the rotation facilitates the reaction. We're beginning to bring the pieces in. For the last example, what I show is a similar idea. So what can we do to alter the random motion of what we're using, right? Oh, I guess I had a concluding slide on this. So the next question is, can one engineer solid-state reactivity to control rotational dynamics? So we made that another structure. So we have a very similar structure here again. And here we have a molecular speed here. So the rotation in this case is fast. If we look at this, the rotation of the group in the center can be fast. And then we bring light, and this trans sort of linear compound becomes bent and clashes now with the rotation there. So this is the fast speed, and this is the slow speed. And again, the way we can tell about that is, by we get our crystals, and we can do spectroscopy to see the changes in the spectrum. Again, there is a change in color in this case. But we can also look at how long it takes to go back, and this is the data that tells us if it is slow or fast. So we're beginning to put all of this together, and I think hopefully what you can see is that there's a lot of work that needs to be done, but I think we're beginning to lay the foundation and really demonstrate that crystalline molecular machines can really be a significant interesting thing to do. And this is basically the message, right? So we need to design and build molecules that have desirable functions as a result of their internal motion or their ability to change shape. That's what we're defining it. And I will finish with this image, right? Because this is the contribution that we have made in our group, that we came up with this concept of amphidynamic crystals as crystals that you can bring, again, structurally encoded order, and that order and the structure also brings motion. So bringing those two together is not trivial, but it's possible. We've shown it. So with that, what I will do is recognize that a lot of people have been involved in doing this research. So this is a list of group members from the time that our group started. And if you look very carefully and have a good eye, you'll recognize here Professor Resendez, who was a member of my group. And if you come to my lecture tomorrow, I can tell you about some of the research that he did, which is extremely cool. So I'm very grateful to all the students, postdocs, and senior collaborators that participated in this research. And I want to thank you also for the invitation, for your attention, and I'll be very happy to answer any questions that you might have. So thank you.
Yeah, thank you for a wonderful talk. Yeah, Doris.
Hi. The last example you gave with the yellow to orange,
Yeah.
is that something you can attenuate? In other words, can you, with the intensity of the light, get different levels of depth of...
Yes. Yeah, if you look at the spectrum that I showed you, show the increase in the absorption as a function of exposure to light, so we can reach different intensities. That's a great question. Yeah. Yes, please.
How many trials did it take you to get a consistent color with the light?
In this case, we knew that it was going to be orange because if we do the studies in solution, we can see it go orange, but it fades away really quickly. So we had an expectation that we would get orange. But if we change the structure of the molecule, we can go more towards the red or more towards the yellow. So we have the ability to tune that a little bit. Yeah.
Any other questions?
Yes, please.
When you talk about machine, one important factor when you think about is efficiency. So I wonder at this level, how did you define, measure the efficiency?
Yeah.
What factor is going to determine efficiency?
I think that is when we're using photochemical activation is very easy because we can measure the quantum yield, right? So the number of events by the number of photons absorbed. When we're measuring other effects, like for instance, we're measuring rotation, we'll make a judgment based on whether we wanted to have, for instance, we wanted to have free motion or we wanted to have a slow motion. And so depending on what the target was. The most interesting phenomena occur when you have when we have free motion or inertial motion. So that rotation in a solid can be almost as fast as it would be in the gas phase. That's really hard to accomplish. So typically we set out to do something like that and we don't get there, so that's not obviously not successful, right? So we need to make some structural modification and then do it. I didn't talk about also the interfacing the electric field of the crystal with the electric field outside. And then we can get a measure of efficiency there, which has to do with the collective response of the molecules. If we have a fraction of the molecule responding, or every molecule responded. But you're absolutely right, so we need to find a metric that determines efficiency. We haven't had an opportunity to measure efficiency the way it is done in biology, for example, because we haven't got to that level of complexity.
Thank you.
Yeah.
Yeah.
Oh, yes.
Yeah, so you mentioned the cis-retinal and trans-retinal isomers. I was wondering if there were, or if there could be any crystalline molecular machines that could be used during like lens replacement surgery in the eye, so like cataract surgery, for example, that could assist in accommodation.
Yeah. So, in my group was very far from anything like that. But I remember I was at a lecture some time ago, where a group of scientists, including medical doctors, what they did is they introduced basically back in the back of the eye a number of photocells that were somehow connected with the optical nerve. And so they were able to restore vision to people who were blind, even though they might have only, say, 4 by 4, 16 pixels. But the amazing thing was that they reported that the images that their brain was able to reconstruct were of very high quality, right? So it was not a 16 pixel image. Their brain reconstructed to make an image that would be many more pixels. So I think what you're thinking is possible.
Okay.
Yeah.
So would that be someone who had sight and lost it? So the brain was already trained?
I think that was the example that they were demonstrating, yes.
So it wouldn't work for someone who was born blind?
That's a really interesting question. I don't know the answer to that, but that sounds fascinating, right?
Oh, yeah.
If the brain is getting that signal for the first time, will it know how to process it and how to, extrapolate what you get, the very little information that you're getting into something that is more meaningful? Yeah, that's a really interesting question. Yes?
So when you're talking about supplying energy to these molecules, that's all light?
At this point, we're using two types of energy, but mainly, so it's either light or it's electrical energy. So we use, I didn't show any of that data, but I think I showed some of it. So we put the crystal typically into two plates, and we introduce an electric field. And so if we can make that electric field an alternate electric field, we can actually drive the rotation by going back and forth. So in that case, when we do that, the energy that is introduced is dissipated at heat, so the crystal gets hot. And the way one can think about it is a little bit like a microwave. And so when you have a microwave, you make the water molecule rotate and that generates heat. We're making a little bit of that also. So we bring an electric field, make it move, and the motion dissipates some heat. So we're learning, little by little how to bring different stimuli, how to deal with the different aspects are required for a machine-like function.
What kind of lights are you using?
Mainly ultraviolet and visible light when we use light. And when we use electric fields, we're using alternating fields that are from a few hertz, a few per second, all the way to gigahertz. That's the nice thing about an electric field, that you can have a huge frequency range. Yeah.
Another question.
Yes.
Adding on to that, I remember you mentioned when it came to the movement, the rotation of the cis keto, that it would change depending on where it was visible light or ultraviolet light, and ultraviolet light would cause it to rotate slower.
Yeah.
I'm wondering if you were to apply infrared light, would that cause it to rotate faster than it would be under visible light?
I think if you were to use infrared light of very high intensity, but very high, you would have a condition where two infrared photons add up to make a UV photon, which is called two-photon absorption. Otherwise, I think, that's a really good question. So if you have well, let's say if you wanted to drive the rotation with light,
Yeah.
right? So the frequencies will fall in the terahertz regime. So you need light that is well. Let me think about that. It's an unusual frequency, but it's a little longer than microwave. Yeah, or shorter than microwave. Yeah, but that's a possibility. If you have a very strong monochromatic beam of light at that frequency, which is about 10 to the 12 per second, you might be able to drive it. That experiment needs to be done.